Well, why is this important? In an editorial in Nature in 2006, the editor said, quote, "In the contentious world of animal research, one question surfaces time and again. How useful are animal experiments as a way to prepare for trials of medical treatments in humans? The issue is crucial, as public opinion is behind animal research only if it helps develop better drugs. Consequently, scientists defending animal experiments insist they are essential for safe clinical trials." In other words, that they are predictive. Whereas animal rights activists vehemently maintain that they are useless. Now, I'm not going to maintain that they're useless, but I am going to maintain that they are not predictive. And let me just delve a little bit further into this word prediction. Let's assume that there are 200 faculty and staff at the University of Wisconsin in the history department. I have no idea how many they are, I just made that up. Well, it's football season, and eventually we will have a bowl championship series, and we'll have a champion in the Division 1A football. And let's assume that the 200 people in the history department all wager on who will be the ultimate number one. Now, it's highly probable that at least one person will get the correct answer. So, can we say then that the history department always predicts the winner of the BCS? Well, no, we can't, because if the history department were predicting, they would only get one pick, not 200. And this is a classic fallacy known as the fallacy of composition. It confuses a collective and distributive forms of a word. But the animal model community is prone to use words in this fashion. For example, thalidomide is a drug that caused birth defects in humans in the late 50s and early 60s. And the drug caused the exact same birth defects in a particular rabbit. Now, some people would say that therefore the animal model predicted thalidomide would be dangerous. The problem with that is that there were over 100 species and strains of animals that were tested, and very, very few of them mimicked the human response. Over 90% of the animals tested did not suffer birth defects from thalidomide. So you really cannot say that the animal model per se is predictive. Neither can you say that the rabbit predicted the response. In order to say that the rabbit predicted this, you would have to see what the rabbit did when tested with other known teratogens, drugs that cause birth defects. Now, if the rabbit mimicked humans 9 out of 10 times, 19 out of 20 times, that's not bad. I would accept that as being a pretty good predictor. But in point of fact, rabbits are very, very poor predictors, and this is just an example where they happen to correlate with human data. Well, the reason that we know that rhino horn is not an aphrodisiac, aside from the fact that I took a whole lot of it in college, is because of the scientific method. Okay, the scientific method is the best method we have of understanding the material world. And we can do studies, and we can actually find things out, and it's really neat. Now, teratogenicity is to see if drugs cause birth defects in humans. Teratogen means birth defects. And there are approximately 1,500 chemicals that cause birth defects in animals. Of those, only 40 have been shown to cause birth defects in humans. Now, we're going to use our little 2x2 table up here, and we're going to calculate the positive predictive value of animal tests for birth defects. And as you can see, it's about 3%. Not stellar. This is a study published in 1990 where 6 drugs that were in clinical trials were studied for their side effects, and then people retrospectively looked back at the animals that were tested to see how everybody compared. And what they found was that animals and humans shared 22 side effects, and we call those true positives. Animals incorrectly identified 48 side effects that in fact did not occur in humans, and the animals missed 20 side effects that in fact did occur in humans. Now, if we use our little box down here in the right-hand corner, we're going to find that the sensitivity of that test is about the same as flipping a coin. And we're going to find that the positive predictive value of that test is 31%. Again, not a number that I would want to hang my life on. This is not my slide. This is a slide from Mark Levin, the former CEO of Millennium Pharmaceuticals. He showed this data at a drug discovery meeting in 2001 in Boston. And his company looked at 28 potential drugs and tested them all on rats to see if it damaged the rat's liver. That's what hepatotoxic means, liver toxic. And in rats, 17 of the drugs were safe and 11 damaged the liver. Levin said, let's test pretty much all of them on humans and just see what happens. Very small doses. They weren't going to kill anybody. And what they found was that of the 11 drugs that were toxic in rats, 6 of them were safe in humans. And of the 17 that were safe in rats, 6 of those were toxic in humans. Now, what Levin concluded from this, and this is, well, it is my conclusion, but it was Levin's conclusion first, is that the rat data was about as accurate as a coin toss. For those of you who don't have a science background in the audience, this is a type of graph that we call a scattergram. And in this study that was published in 2002, the authors compared animal bioavailability of drugs, which is on the x-axis, with human bioavailability of those drugs, which is on the y-axis. And they looked at primates, rodents, and dogs and compared that data with humans. And as you can see, there is absolutely no correlation whatsoever in this data. This is a meeting that I attended 1 year ago this week that was sponsored by the CBI, and it was comprised, it was a really neat meeting. It was comprised of, it was a closed meeting, and I only got invited through a fluke. But it was comprised of people from the pharmaceutical industry, the NIH, and the FDA. And like I said, it was a closed meeting, and it was pretty neat because pretty much everybody who was in the audience was also a speaker, again, except for me. And the premise of this meeting was that animal models are not predictive for humans. Okay, that was the given. And it was, again, it was neat because the pharmaceutical companies came together and shared data. So Pfizer's up there saying, we don't have anything that predicts what a drug is going to do in humans. And it's predictive ADMET. A stands for absorption, D for distribution, and so forth. And these are the parameters that we try to use animals to determine. Okay, ADMET is what it's called for short. So Pfizer gets up and says, here's what we're doing. It doesn't work, but, you know, does anybody else have any ideas? And then the scientists from Merck get up there and say, we know animal data doesn't work. We're trying this computer method, and so on, and so on, and so on. But it really kind of floored me after 10 years of going around the country and saying the animal model is not predictive to have representatives of NIH, FDA, and, you know, the big pharma up there agreeing with me. I wondered what I was going to do when I went home. Well, let's talk a little bit about diseases. Now, I've been focusing on drugs, and drugs are very easy to measure. It's a very easy thing to quantify. You can get very nice statistics from them, etc. But let's look at some diseases. Now, this is a quote from a textbook in 1980, but this is a quote that summarizes hundreds of studies. Okay, so this quote by itself would be considered anecdotal, but the fact that it summarizes hundreds of studies, I think, speaks for itself. And it says, for decades, the clinical observation of an association between cigarette smoking and bronchial carcinoma was subject to unfounded doubt, suspicion, and outright opposition, largely because the disease had no counterpart in mice. There seemed no end of statisticians craving for more documentation, all resulting in the fateful delay of needed legislative initiative. The tobacco companies rode that puppy for decades before they had to put that little label on the side of the cigarette carton. This is from a textbook in 1948, and again, this summarizes hundreds of studies. And the authors say, there is no satisfactory evidence that the incidence of atherosclerosis bears any relation to the concentration of cholesterol in the blood. Now, in the old days, you know who used to advertise cigarettes? Doctors. I have a whole slideshow of ads showing doctors recommending menthol cools or whatever. And this is very similar, because today, if you have a heart attack, we strongly recommend that you cut back on the fat in your diet and try and lower your cholesterol. In the 40s and 50s, we put you on a high-fat diet. Okay? That's not good. And this is a study, again, that summarizes scores of studies that were done on asbestos. This was published in 1965. It says a large literature on experimental studies has failed to furnish any definitive evidence for induction of malignant tumors in animals exposed to various varieties and preparations of asbestos by inhalation or intratracheal injection. And that kept asbestos on the market for a long time. Okay, a little more recent example, this is HIV. This is what some of the blood values do in humans when humans are infected with HIV. And as you can see, it's either completely opposite or not correlated to what the same blood parameters do in chimpanzees. And just FYI, the scientific community doesn't even use chimpanzees for HIV research anymore, despite the fact that they are our closest relative. Now, Van Zutphen, I think that's how you pronounce his name, is an animal modeler himself. And he wrote an article in 2000 in Comparative Medicine and said that animal models can be used to find out which genes cause diseases in humans. Okay, that's fine. Then he goes on to say this. However, results to date suggest that the predictive value of a candidate gene established in such an animal model is rather low. Thus far, only few genes have been allocated as causative factors for corresponding disorders in man. In fact, it can be questioned whether the use of animal models is the most effective way to detect candidate genes for complex human disorders. Due to the complexity of the genotype-environment interactions, the pathways that lead to an aberrant phenotype often differ between man and animal. And this is an article from 2004, EMBO reports. It says knockout experiments in mice in which a gene that is considered to be essential is inactivated or removed are widely used to infer the role of individual genes. That's true. In many such experiments, the knockout is found to have no effect whatsoever, despite the fact that the gene encodes a protein that is believed to be essential. In other cases, the knockout has completely unexpected effects. Furthermore, disruption of the same gene can have diverse effects in different strains of mice, not different species, different strains. Such findings question the wisdom of extrapolating data that are obtained in mice to other species, like humans. In fact, there is little reason to assume that experiments with genetically modified mice will necessarily provide insights into the complex gene interactions that occur in humans. Okay, so since we're at a liberal arts college, let's bring in a little liberal arts and quote Lewis Carroll. And Lewis Carroll says, through Humpty Dumpty here, he says, "When I use a word," Humpty Dumpty said in a rather scornful tone, "I mean just what I choose it to mean, neither more nor less. But the question is," said Alice, "whether you can make words mean so many different things." I've given you a definition of what prediction means. Again, I think that's really self-evident, but I've nonetheless supplied you with definitions. Let me tell you what prediction does not mean. It does not mean that the animal model gives us a general idea, or points us in the right direction, or makes us think, or lets us know if we were on the right track. Occasionally getting the answer right is not the same as predicting it. Now, you frequently hear people say, we tested 25 species and one ended up giving the same results as humans, therefore it was predictive. No, it wasn't. Not unless you can show me studies of 20 or 30 tests with different drugs that animal also got right. And you'll also hear people say for that for any given drug, an animal can be found in retrospect that mimicked human response. Therefore, the animal model per se is predictive. And again, this is like saying the history department always predicts the winner of the BCS.